Hello Ecology Lab. In this video I'm going to show you how to plot rarefaction curves that we did in lab number 6 with our bird abundance data. So remember what rarefaction does is it's a way to deal with the fact that you might have different diversity, so you might have different species richness or something like that in two different samples. So we're going to compare two communities and we want to see which one is more diverse. But the problem is, right, the diversity that you quantify is going to be affected by the sample size or the sampling effort. Because if you get more species in your sample, or if you get more individuals in your sample, you're more likely to get rare species, so you're more likely to get more species and higher diversity. So we did rarefaction with a program called EcoSim. And this is the output from EcoSim that I've imported into Excel here. So remember what EcoSim does is it takes 1,000 simulations. And so we have our community that's, I think this is breeding bird data from the whole state of Colorado. And so we had 62,000, actually I think we had something like 65,000 individuals sampled. And so what this does is we're going to compare this to another community. And what EcoSim does is it randomly pulls out individuals from that sample of 65,000 birds, and then it figures out how many species you have. So it basically determines species richness of those subsamples. And you can do this over a whole bunch of levels of abundance, and then you get to see, on average, how many species you get. So for instance, the first thing that EcoSim does is it chooses one individual out of that sample of 65,000 individuals. And of course, the diversity is 1 because we get one species, right? The median is also 1. There's no variance, and then the 95% confidence limits are also, range from 1 to 1, right? But if we pull out a random 1,273 individuals from that 65,000, then on average we get 130.712 species represented, right? Of those 1,000 simulations, on average we get about 130 species, and importantly, the 95% confidence limits range between 121 species and 139 species. If we go way down here and we pull out 50,861 individuals from the 65,000 individuals, we get 217.8 species on average, and our confidence limits range between 215 species and 221 species. Okay, so this will allow us, if we plot rarefaction curves for multiple communities, then we can compare diversity directly. One thing EcoSim doesn't do is it doesn't plot the graphs for you. So I've exported the data from EcoSim, I've put it into Excel, and then I save this as a tab delimited text file, which I can pull into R. So if I go into RStudio here, up here in the upper left, I've written some code to plot these curves. So the first thing is you have to show R where the data are. So you can run this line set working directory and then specify the path to where your files are. Another way to do that in RStudio is you can set the working directory. So I can click those buttons and I can navigate to this. I have this on my C drive in a folder called R and a folder called Rarefaction Lab. And here's my data right here, my Breeding Bird Survey 1978, Breeding Bird Survey 2018. So what I'm going to do is click on More here, and I'm going to set as working directory. Okay. Now I'm going to read in the data, so I'm going to run this line. So I'm going to make a variable called BBS 1978 for Breeding Bird Survey. Use that assignment operator. And I'm going to use the read.delim function and read the file, the tab delimited text file that's in my folder over here. So I'm going to run that line, Alt Enter. Okay, great. And we can see in my data set up here, now I've got that variable called BBS 1978, 50 observations of 6 variables. Okay, so 6 columns in the EcoSim output. I'm going to do the same thing here, add the 2018, and now I'm just going to plot it, right? So I'm going to use the plot function. If we use help, so I'm going to go down to my console window, and I'm going to put help, and in parentheses, plot. And you can do this with any function, and it'll bring up, oops, I misspelled it, help plot. You can do this with any function, and it'll bring up the help over here of what the arguments are in that function, right? So plots, we're going to plot our x and then our y. We've got different types of plots, x labels, y labels, all of those sorts of things. Right? So in here, I'm using plot, and I'm first going to plot my 2018 data. So I'm going to plot BBS 2018 and the column labeled abundance. Right? If we look back on here, the abundance here, this is the number of individuals in the sample. Then I'm going to plot the average diversity on the y-axis, which is the number of species. Okay, so my x is BBS, $ tells it what column to choose, and then abundance. My y is going to be the same thing. Oops, I'm up here. My y is going to be the average diversity. Type equals L, that means I want a line graph. LWD, line width 2, means my line's going to be a little thicker. For the color of the line, I chose a color called steel blue. You can Google colors in R and you'll see a whole list of different colors. My X label, I'm going to call that abundance. My Y label is going to be number of species, and my main label I'm just calling Colorado Breeding Bird Survey 1978-2018. Okay, so let's run that. Alt-Enter, and there's my plot. All right, so we can see my abundance on the x-axis, my number of species on the y, and we can see that curve. Now I'm also going to add the 95% confidence limits. So I'm going to do the same thing. My x is abundance. My y now is going to be my low confidence limit. Same type line graph, but I'm going to use for LTY as line type. The default is a solid line, but I'm going to make it a dashed line. So let's run that code. And there's my lower confidence limit. I'm going to do the same thing for the upper confidence limit. There we go. And now I'm going to add the 1978 data. So I had the same thing here. I use the lines function to add another line to the graph. But here I'm using 1978 abundance as the x, average diversity as the y, and I'm going to change the line type to, or the color to coral 2. And there we go. So there's the average diversity, and I'm going to add my confidence limits, the lower one and the upper one. Okay, now the only thing to add here is a legend. So legend, first you give it the x and y of where you want the legend plotted. So on my x-axis, I want to put it down here in the lower right. So I'm going to put it at about 50,000 on the x-axis and about 50 on the y-axis. So I'm going to put it right around there. My legend is going to equal, and my labels here, I'm going to put C and then parentheses, and I'm going to label them 2018 and 1978 for the order that I put them in there. And my colors, steel blue and coral 2. Line type 1, 1, that means a solid line, and line width is going to be 2 for each one. So let's run that, and there we go. So now you can just export this plot, save it as a JPEG or something, and include it in your lab report. This is very similar to the Whittaker plots that we've done in the past.